Good morning, good afternoon, everyone. Before I start this presentation, I would like to thank the organizers for giving me the opportunity to participate in cancer webinar. I'm pleased to share with you our published work entitled Biological Impact of Advanced Glycation and Products on Estrogen Receptor-Positive MCF7 Breast Cancer Cells. This research work was partly conducted at KAIMRC, King Abdullah International Medical Research Center in Riyadh. To give a brief presentation of KAIMRC, KAIMRC is a part of three entities considered as centers of excellence in Riyadh, where numerous biomedical research projects have been conducted using state-of-the-art and cutting-edge facilities. The two other entities are University of Health Sciences and the Hospital King Abdulaziz Medical City. Similar campuses composed of three entities are found in other regions of Saudi Arabia. The main one is in Riyadh. Breast cancer is the most common cancer in women worldwide. In 2020, it was estimated more than 2 million new cases and 780,000 breast cancer-related deaths occurred worldwide. Based on the global cancer statistics, Global CAN 2020, breast cancer incidence was reported to be the highest in developed countries, including Australia, New Zealand, Europe, and US, and the lowest in developing countries, including Africa and South Central Asia. This schematic diagram presents the main risk factors resulting in breast cancer etiology, such as family history, including familial breast cancer, corresponding to 15–20% of the cases, and 5–10% hereditary, concerning many patients presenting BRCA1 and 2 mutations. We have also age, gender, reproductive history, and also environmental factors and lifestyle, leading to sporadic breast cancer, the majority, corresponding to 70–80% of the cases. At the molecular level, breast cancer is a heterogeneous disease classified into different subtypes based on hormone receptor expression. Along with the expression of progesterone receptor, estrogen receptor-positive breast cancer is the most common subtype, corresponding to 65–75% of all breast cancer subtypes and are known to be poorly invasive, followed by human epidermal growth factor receptor HER2+, corresponding to 15–20%, and the highly invasive and aggressive triple-negative breast cancer, which corresponds to 15% of the cases. Thus, this heterogeneous malignancy is characterized by a highly diversity of morphological, molecular, and biochemical features influencing the disease progress, prognosis, and the response to therapeutics, including HER2-targeted drugs, hormone therapy for ER+, or combination of all these treatments. I have to mention that due to the lack of hormone receptor and HER2 expression, the anti-cancer therapy for the triple-negative breast cancer remains challenging. Now moving to diabetes, a severe metabolic disorder reported to enhance breast cancer risk. This heat map of the global diabetes prevalence shows that the North, East, and the South of Africa, Arabic Peninsula, South Asia, North and South of America are among the top countries with a high percentage of prevalence, corresponding to more than 12.5%, as reported by International Diabetes Federation in 2019. Diabetes, also known as diabetes mellitus, concerns two types. Type 1, a multifactorial disease with a strong genetic component, causing autoimmune destruction of insulin-producing pancreatic beta cells, which develops at a younger age. While the type 2 diabetes mellitus is a lifelong chronic disease commonly diagnosed in aging population. Worldwide, over 450 million people have diabetes, with an estimated increase reaching 642 million diabetic patients by 2040. The treatment of diabetes costs $1,000 per patient. However, the cost considerably increases when the patient presents diabetes-related complications, reaching $25,000 to $48,000 per year per diabetic patient. Therefore, there is an urgent need for biomarker identification and tailored therapy development in diabetic patients diagnosed with breast cancer. Regarding diabetes-related risk factors, this schematic diagram describes the main metabolic consequences of a mismanaged diabetes characterized by hyperglycemia, hyperinsulinemia, and hyperlipidemia, leading to breast cancer progression. Among these metabolic consequences caused by hyperglycemia, it occurs activation of transcription factors such as nuclear factor kappa B, responsible for inflammation, activation of signal transducer of activator of transcription site 3, responsible for cancer onset, development, and progression, and hypoxia-inducible factor, HIF1-alpha, activation resulting in a tumor angiogenesis in low oxygen condition. Caused by hyperinsulinemia, it occurs a decrease in hormone-binding globulin, which augments the amount of free hormones, including estrogen, and thus increase estrogen receptor signaling, and subsequently enhance the mammary epithelial cell response to the estrogen. These dysregulated metabolic pathways result in the stimulation of breast cancer cell events, contributing to breast cancer development and progression through increased cell survival, proliferation, migration, and invasion metastasis. In hyperglycemic conditions, various biological macromolecules, including proteins, lipids, and nucleic acids, undergo a non-enzymatic glycation reaction named as the Maillard reaction, resulting in intermolecular cross-linking between nucleophilic amino group of these macromolecules and the reactive carbonyl group of sugars to form advanced glycation endproducts, AGAs. During metabolic reaction of the glucose, with the time it becomes clear that these adducts undergo slow rearrangement to form AGAs. Among the reactive carbonyl metabolites, methyl glyoxide, a reactive decarbonyl metabolite of glucose, has received considerable attention as the most reactive AGA precursor. Despite it is a non-enzymatic reaction, proteins undergo this glycation modification more frequently than other macromolecules, with substantial functional consequences, including altered enzyme activity, increased immunogenicity, decreased ligand binding, increased cross-linking of extracellular matrix proteins causing stiffening, or affects cell extracellular matrix interaction. Due to the high risk of production of AGAs in type 2 diabetic patients, hence it is important to know what are the biological effects of AGAs on ER-positive breast cancer cells. Therefore, to answer to this question, we established an experimental design. Immortalized estrogen receptor-positive MCF7 breast cancer cells were exposed to various concentrations of homemade glycated bovine serum albumin generated using methyl glyoxide, referred to as MG-BSA AGAs, or exposed to non-modified BSA, in order to reveal the impact of the non-enzymatic glycation of the serum albumin, the major plasma globular protein. Thus, to assess the biological effects of AGAs on breast cancer cell proliferation, migration, and invasion, in vitro cell-based assays were applied. An investigation of the molecular mechanisms was done focusing on RAG, the main receptor for AGAs, and on ER-alpha and ER-beta gene protein expression using RT-qPCR, Western blot, and FACS. Signaling pathway analysis was performed using phosphoprotein array. Our first results show that at all concentrations used, the non-modified BSA had no effect on MCF7 cell proliferation and directional migration. However, increasing concentration of AGAs resulted in the modification of cell proliferation and migration in a dose-dependent manner, with a peak of stimulation reached at 100 micrograms per ml of AGAs compared to the control, the untreated cells. This observed bell-shaped curve is an indicator of the observed AGA stimulatory effect that may occur through a receptor, such as the main receptor for AGAs named RAG. At the highest concentration of AGAs, at 200 micrograms per ml, a significant decrease in MCF7 cell number and viability was associated with the induction of apoptosis, indicated by the detection of cleaved caspase 3 observed in the cells exposed to 200 micrograms per ml of AGAs only. However, the untreated and BSA-treated cells, no cleavage of caspase 3 was observed. The housekeeping protein gap DH was used to confirm the equal loading of the protein amount. After performing MCF7 cell invasion assay using matrix-coated inserts in the presence of high concentration of AGAs, we showed that AGAs did not affect MCF7 cell invasion. The conditioned culture media were collected for gelatin zymography for the assessment of the gelatinase activity of the MMP2 and MMP9, whose active form degrades the extracellular matrix protein for the cell invasion to occur. We showed that when tested at 100 micrograms per ml, AGA significantly increased the production of pro-MMP9 corresponding to its inactive form, which explained the absence of AGA's effect on the MCF7 cell invasion. In contrast to AGA's induced pro-MMP9 production, no change of the activity of pro-MMP2 inactive form was observed in all AGA's concentrations tested. In addition to the increased production of MMP9 shown in the bar graph, a twofold increase in MMP9 transcript was also observed in AGAs-treated cells, while no MMP9 upregulation was observed in BSA-treated cells compared to the basal level monitored in the control. Concerning the investigation of the signaling pathways induced by AGEs, the MCF7 cells were exposed to various concentrations of AGEs and non-modified BSA for 10 minutes incubation. Using Western blot, we firstly assessed the biological impact of AGEs on the activation of the main signaling protein, the mitogen activated protein kinase phosphoextracellular signal regulator kinase ERK1/2. This representative Western blot shows that no overphosphorylation of ERK1/2 was observed at all BSA concentrations compared to the basal level of phospho-ERK1/2 detected in the control, the untreated cells. However, at 50 and 100 micrograms per ml of AGEs, an obvious increase of the phospho-ERK1/2 expression level was observed. At the highest concentration of AGEs, 200 micrograms per ml, associated with its significant inhibitory on MCF7 cell proliferation, a decrease in ERK2 phosphorylation level was observed as well. Further signaling pathway investigation was performed testing both AGEs and BSA at 100 micrograms per ml. After MCF7 cell exposure to AGEs and BSA tested at 100 micrograms per ml, the cell isolates were subjected to phosphoprotein array composed of multi-immunoblots which contain a large panel of antibodies targeting the main phosphorylated signaling proteins, including phosphoforms of ERK1/2 and MKK1/2 of the MAP kinase pathway, and CREB1 and protein kinase C family. This bar graph summarizes the expression level of phosphoproteins assessed in AGEs and BSA-treated cells. For both conditions, relative expression levels were normalized to the control, the untreated cells, corresponding to 1. No changes in the phosphoprotein level were observed in cells treated with the non-modified BSA, except for the MKK1/2, CREB1, and the protein kinase C epsilon, whose their expression levels were increased. As previously mentioned, a slight increase in the phospho-ERK1/2 expression level was observed, and MKK1/2 and CREB1 expression was also induced by the addition of AGEs at a higher expression level detected than in BSA-treated cells, while a clear inhibition in the phosphorylated form was detected for protein kinase C family members, adenosine alpha and MSK1 in AGEs-treated cells. Using similar application, we also studied its biological impact on highly aggressive triple-negative breast cancer using MDM-B231 cells. After 10 minutes of incubation as compared to the control, AGEs clearly increased the phosphoform of most of the signaling proteins, including ERK2 and the P70S6K1, while BSA did not modulate their expression. Indeed, AGEs increased by 7.5-fold most of the phosphoprotein expression levels. Interestingly, AGEs upregulated by 25-fold phosphorylation of the P70S6K1, ribosomal protein involved in protein synthesis. These interesting findings suggest phospho-P70S6K1 as a potential biomarker for highly invasive breast cancer patients diagnosed with diabetes. However, regarding ER-positive breast cancer cell line, MCF7, a slight inhibitory effect of AGEs was observed on the phosphorylation of P70S6K1, while BSA had no effect. Regarding the other key signaling proteins such as STAT3, JNK, MAP kinase, GSK3-beta, similar decreased expression level of the phosphoform of these signaling proteins was detected in both AGEs and BSA-treated cells as compared with the untreated cells, the control. Then we monitored the protein expression levels of RAG, ER-alpha and ER-beta in MCF7 untreated cells and in cells treated with BSA and AGEs at 100 micrograms per ml and at different incubation time periods. At increasing incubation time, AGEs significantly augmented RAG expression level, the black bars, while BNA had no effect as compared to the control. Regarding ER-alpha, the white bars, although a 30% increase in ER-alpha expression level was monitored in untreated cells and in BSA-treated cells, AGEs significantly increased by 60% ER-alpha expression level after 72 hours of incubation. Regarding ER-beta, the gray bars, which exerts anti-proliferative effects, a 50-60% increase in ER-beta expression level was observed in untreated cells after 40 and 72 hours of incubation, and in BSA-treated cells after 24 and 48 hours of incubation. However, cell exposure to AGEs resulted in a significant 20% decrease in ER-beta expression after 24 and 48 hours of incubation, and 20% increase in ER-beta expression levels in AGE-treated MCF7 cells after 72 hours of incubation. The respective upregulation of the protein expression levels of RAJ, ER-alpha and ER-beta induced by AGEs, observed after 72 hours of incubation, was confirmed at the gene expression level using RT-qPCR compared to those monitored in BSA-treated cells and untreated cells. Playing a key role in AGEs' biological effects, we wanted to know whether the stimulatory effects of AGEs resulting in the increase of MCF7 cell functions were correlated with the increase in RAJ expressed on the cell surface of the MCF7 cells. After 72 hours of incubation, the cells were harvested, incubated with the anti-RAJ monoclonal antibody conjugated with a fluorochrome, FITC. The cell exposure to AGEs resulted in a decrease of RAJ expressed on the cell surface, while the amount of RAJ detected inside the cells after cell membrane permeabilization was almost doubled as compared to the intracellular RAJ amount detected in the control, the untreated cells. Knowing the important function of ER-alpha in the hormone-dependent breast cancer development and progression, we wanted to check if there is any change in ER-alpha expression at the nuclear level. Using confocal microscopy after different incubation times of cells untreated or treated with either 1 microgram per ml of BSA or AGEs, these representative photomicrographs show the cytoplasmic and nuclear localization of ER-alpha in the MCF7 cells. It is only in AGE-treated cells that we can clearly see intense staining of ER-alpha in the nucleus and especially into the nucleoli, sites of high transcriptional activity. The increased ER-alpha related transcription activity was confirmed in AGE-treated MCF7 cells using an ER-alpha transcription factor activation kit. No change of this transcription activity was observed in BSA-treated cells. To confirm the key role of RAJ in AGEs-induced MCF7 cell signaling, including ERK1/2 phosphorylation, RAJ loss of function was performed using anti-RAJ monoclonal antibody along with the ASO-TAP control IgG1. As shown on this representative Western blot, RAJ loss of function impeded AGEs-induced ERK1/2 phosphorylation, while AGEs-induced ERK1/2 phosphorylation was observed in IgG1 pretreated cells. Similar biological outcome was observed in regards with RAJ and ER-alpha. A significant increase of both protein expression levels induced by AGEs was observed in IgG1 pretreated cells, while no change of their respective expression level was observed in anti-RAJ antibody pretreated cells. These results demonstrate the suppression of AGE stimulatory effect in MCF7 cells after RAJ loss of function. In conclusion, MG-BSA AGEs at 50-100 micrograms per ml exert a dose-dependent stimulatory effect on MCF7 cell proliferation and diagonal cell migration, but do not affect their weak invasive capacity. Signal transduction pathway analysis suggests that AGEs might contribute to breast cancer development by activating both MAP kinase and the CREB1 transcription factor. AGEs upregulate the receptor for AGEs, RAJ, and ER expression at the protein and transcript levels in MCF7 cells in a RAJ-dependent manner. Therefore, targeting RAJ expression may be a promising approach to prevent the development of ER-positive breast cancer among patients with diabetes mellitus. This study was published in a high-impact factor journal, Biochemical and Biophysical Acta, Molecular Basis of Disease, in 2017. In the same journal, we published in 2015 Advanced Glycation Endproducts Increased Proliferation, Migration, and Invasion of the Breast Cancer Cell Line, MDA and B231. In conclusion of this second publication, we demonstrated for the first time that MG-BSA-AGEs exert a stimulatory effect in a dose-dependent manner on an invasive and non-hormone-dependent breast cancer cell line, the MDA and B231 through RAJ, as compared to non-modified BSA and to the control the untreated condition. We also highlighted a concomitant increase of P76K1, which might be a new biomarker of the invasive triple-negative breast cancer subtype and a promising target drug to fight against breast cancer in diabetic patients. I would like to acknowledge the active contribution of cell and gene therapy team members and core facility from KAIMRC and PhD students and research scientists from Manchester Metropolitan University in UK. Thank you for your attention.